So it's a pleasure to have as our next speaker, Raymond Goldstein. Ray's going to tell us about anti-diffusion in various biological things. Ray, please.
Well, thank you so much for the organizers for the opportunity to be here. I confessed when invited that I'm not sure I really work on anti-diffusion, but then I think I've come to realize that it's not quite clear what the definition of anti-diffusion is. And so that makes it a little safer for me to talk. So today I want to tell you about some work that we've recently finished in my lab, which is 50 meters that way, 100 meters that way, in DAG. It's a problem that is very interdisciplinary, and I thought I should take some time in explaining what we did to kind of explain the intellectual origins of it, because I think it's a nice case study in how talking to people from other disciplines can somehow give you interesting ideas. So the work involves my postdoc, Praneet Prakash, where's Praneet? Raise your hand. There he is, back there, who's pioneered all the experiments that I'll be describing and a lot of the interpretation. And working jointly as co-first author on this paper is Yasa Beg, who's, I think, online. Hi, Yasa, if you're online. He's an undergraduate, finished at Duke, and has been spending a year doing a master's funded by the Marshall Program in the Cavendish lab with us as the supervisor. And the work actually derives originally from some preliminary important experimental work by Francois Potisserf, who was a PhD student with me some years ago, who's now in University of Rennes, and to establish proof of principle for what I'm going to describe. So it's a problem whose origins can basically be traced back to a very interesting paper that I learned about soon after I got here in Cambridge, due to some of our eminent plant scientists down the road here in Cambridge. So this is a bit far from many of your expertise, but let me just try to explain what's going on and give you an idea of the motivational questions. So back in 2005, Alison Smith's group in Cambridge Plant Science published what turns out to be a landmark paper in which they, in the usual way of declarative titles in biology, established this, that algae, green algae in the ocean and freshwater environments, acquire vitamin B12 by a symbiotic relationship with bacteria. And you might say, who cares? But actually there was a very interesting question that had been around for an awfully long time in sort of marine ecology, which was maybe more general than vitamin B12, but it was cast in the language of vitamin B12, which is there are various kinds of organisms on Earth, some of which need vitamin B12 to function, like us. Some of them don't need it. Some of them can produce it themselves, and some that need it get it from things they eat. And the question was, where do the algae in the ocean who need vitamin B12 to live get it? And although it may seem strange, the prevailing point of view prior to this paper was that there was enough vitamin B12 just present ambiently in the ocean from all the dead organisms that gave it back up to the medium to supply what was ever needed by these organisms by diffusion. Now, it doesn't sound quite plausible, I think, even if you just try to put in elementary numbers, but this group basically decided to investigate this more clearly. And what they discovered was that many algae, which are unable to synthesize vitamin B12 and do depend upon the environment, actually get it because there are bacteria physically associated with them that synthesize the vitamin B12 and give it to them. And they did this by a whole bunch of clever experiments and studies of the existing literature. And the basic point was that the bacteria appear to benefit from the algae through products of photosynthesis, and interestingly, the production of B12 by the bacteria is upregulated in the presence of algae. The algae give off things which stimulate the bacteria to produce B12, and the bacteria then give that to the algae. So when I first heard about this, because we were working on problems involving collective dynamics in active matter and things like that, I immediately thought there were a bunch of really interesting questions here that biophysicists and fluid dynamicists and others like us might think about. For instance, the sort of spatio-temporal dynamics of this symbiosis. You've got microorganisms in the ocean, in a turbulent environment, being pushed around. How do they find each other? How do they stay together? What is the dynamics of this exchange when the algae reproduce and go off in a new direction? Do the bacteria follow them? A whole bunch of sort of elementary questions about this symbiotic relationship. And so later we did a little bit of work, mostly with Francois, on trying to understand some aspects of it. But we always thought it would be interesting to have a kind of model system where there was some kind of metabolite exchange between two members of two different kingdoms of life, like algae and bacteria, to try to have a kind of reductionist approach to this symbiotic problem. So I hope it's clear how we go from this very general, very specific problem of vitamin B12 to a general question of how on earth to study spatiotemporal dynamics of symbiosis. Okay, so then what on earth could you use as some kind of more convenient exchange metabolite than vitamin B12? And here I want to point out an article that I wrote some years ago in Physics Today that was basically about lessons from the old literature. So I wrote this article because on a number of occasions, including in my own work, I, like probably most of you, came across situations in which papers were published that seemed not to be aware of, I don't know, some 100-year-old result where it was all first done. And I thought this was silly, and I should kind of admonish the community, myself included, to keep reading the old literature, not only to sort of be doing due diligence and things like that, but also to get inspiration. So my paper was divided into sections: learn about connections between fields, like we're doing here, do due diligence when you claim something's new, and interestingly, discover inspirational cleverness. And I'll give you an example of that, which is important here. Also, actually, to read the papers that you cite instead of just citing them, because actually, you know, near the end, there are interesting things that nobody ever tells you about. And also to read widely and to quote the motto of the Royal Society, don't trust anybody's word for it. Okay, so the example that I really, really like about getting inspirational cleverness covered is due to Theodor Engelmann. How many of you have ever heard of Engelmann? He's super important in the biology world because in the early 1880s, he made two really important discoveries. The first was he found the first example of what we might call oxygen taxis, chemotaxis toward oxygen of microorganisms. And this was done by actually having a leaf exposed to sunlight, doing photosynthesis, giving off oxygen, and certain bacteria that were found in putrefying matter swam toward this leaf. So one deduces they are oxygen tactic. That was the first discovery, so the first example of oxygen taxis. But then the super clever thing that he did was to figure out a way to actually measure what is called the action spectrum of photosynthesis. So you may know that the photosynthetic apparatus in photosynthetic organisms is sensitive to different colored lights, mostly in the blue and a bit in the red. And there was a lot of discussion in that period of time about what is the exact response of that photosynthetic apparatus to different wavelengths of light. And so he came up with the following, I think, brilliant idea. He asked Carl Zeiss, the microscope manufacturer, to produce this object. It's a microscope which has a little mirror at the top, which would direct sunlight down onto the microscope stage. So instead of, I don't know, a candle or whatever might have been used at that time, you actually use sunlight. And then just before the light hits the microscope stage, you put a prism, as Newton would have done. And you split the sunlight into a spectrum, which you then shine on the microscope stage from blue to red. And you put on the microscope stage a long filamentous green alga, centimeters long, maybe a millimeter or so across, such that parts of it are illuminated by blue and parts by green and parts by red. And then you put in the fluids surrounding the alga those same oxygen-tactic bacteria. And they just gather around in proportion to the rate at which oxygen is being produced. And you just read off the response of the photosynthetic system. So it's essentially the first example of bacteria as biosensors. And it's even better because it turns out when you look at the spectrum, there are certain lines. I'm sure you've all heard of Fraunhofer lines. These are absorption lines from heavier elements than helium and hydrogen that are in the sun. They show up as dark lines. They're sitting there on your microscope stage. You know the wavelength of them already, so you calibrate it that way. So this was really interesting, and we actually reproduced this device in the lab, and it was kind of interesting to see. And there are actually interesting questions even in interpreting this experiment about diffusion and chemotaxis and all of that, but I'll leave that for another time. But this suggested to me that this was actually the right kind of system to use in the sense that we've got bacteria and algae, but oxygen is something we can turn on and off by just shining a light on the system. So the idea that we had was essentially to develop a co-culture of bacteria and green algae. The bacteria are oxygen-tactic, and the algae produce oxygen by photosynthesis by a light that we can turn on and off. And then the only question is what species to use. And you could use many. But we had a lot of experience with the bacterium Bacillus subtilis, which is, as I'll explain, has been very important in the history of active matter for the various phenomena that it exhibits. It's well studied from a whole variety of perspectives. It's often used to study sporulation and all sorts of issue of biofilm formation. Most importantly, especially here, there are various fluorescent strains which we can take advantage of. Jim Hasselhoff in Plant Sciences kindly gave us a yellow fluorescent version of this, which is very useful. Here's an example of what it looks like. The cell, this is in the course of cell division. The cell body is a few microns long. It has many flagella that come out from all over the cell body. It's called paratrichously flagellated. And it swims along, sort of like E. coli, if you're familiar with that, where it executes a kind of meandering or stochastic sort of random walk as it swims through the fluid. Now, for the alga, instead of using something that's just long and skinny and macroscopic in a sense, we wanted to use Chlamydomonas reinhardti. Chlamydomonas is another model organism for the study of many things like photosynthesis, phototaxis, and also here you see a picture of it. It's about 10 microns across with two of these flagella or cilia that are just like the ones that are in your respiratory system and elsewhere in your body, constantly pushing fluid around by a breaststroke motion. So this is a wonderful and well-studied and easy-to-grow model organism. In particular, there are interesting motility mutants, such as those that are missing flagella. And that's useful if you want to just, say, turn off the motility of the green algae and say, no, let's just think of these as little sources of oxygen that come from photosynthesis. So we settled on this, and the question was basically, could you actually get these things to happily grow in the same medium? And then we tried to do an experiment along the lines of Engelmann. So that's what Francois Potesserf did in his PhD at the very end. He actually developed this co-culture and was able to show the first examples of something which I will then focus on in the later part of this talk. So let me just spend a little bit of time talking about what we know about collective behavior from the work of Bacillus subtilis, because it's actually very important for interpreting what we'll eventually see. So long ago, 20 years ago now, when I was at my prior institution, the University of Arizona, and working very closely with John Kessler there, we studied, as many people have studied, a phenomenon called bioconvection. The way you study this in the lab is you have a petri dish with a drop of suspension of bacteria. You've got a ring light somewhere down here, so you're doing dark field illumination, and a camera here on a long bellows, so you get a macroscopic view. And what you see over the course of an hour or two is the development of a very complicated sort of convective turbulent pattern, which is driven by oxygen taxis. So these bacteria, Bacillus subtilis, are called obligate aerobes. They require oxygen to live. If you put them in a suspension of fluid with an air-water interface, they will consume the oxygen that's naturally dissolved in the fluid. More oxygen is coming in from that interface. That creates a gradient going down into the medium. They climb that gradient up to the surface to get more oxygen. However, they're about 2 or 3 or 4% denser than the water that they're swimming in. So having accumulated at the top, you have an unstable stratification, sort of like a Raymond-Taylor kind of system. And so you end up with plumes of bacteria that fall down, but still the oxygen gradient points up. And so you end up with a persistent dynamics of bioconvection. And it's a little bit like the Bénard convection. There's a mapping between the two problems. What is important in this dynamics is not the bioconvection itself, but actually the geometry of this drop. It's a sessile drop sitting on the surface, so it's got tilted edges, the usual kind of shape. And after a while, when all of these plumes kind of settle down, you end up with the bacteria kind of flowing to the edges of the drop. and they accumulate there at very, very high concentrations. And what we discovered with John was something which is now called bacterial turbulence, and Sriram mentioned this before. He made the usual comments that, well, I'm not quite sure whether it really should be called turbulence. However, speaking of astrophysics, which is represented in this audience, my friend, the late Ed Spiegel at Columbia University, when I first told him about this and said, well, I get a lot of pushback for calling it bacterial turbulence, he said, no, no, no. The Latin root of turbulence is turba, which is crowds. Turbulence refers to the milling about of crowds. So this is turbulence. Everything else, I don't know what it is. So thank you, Ed. Right, so this is what you see. Here's a field of view of about 300 microns. So the pendant sessile drop is coming down. That's the contact line with the plastic petri dish right here. And this is a real-time movie of what you see. So behold, bacterial turbulence. Remember, the individual bacteria are 2 or 3 or 4 microns long. So these swirls and eddies that you see are on a length scale that's much larger than the individual bugs. I'll say a little bit more about this. I'm not going to spend the talk on this because it's history. But the most important thing is that in that left-hand image, you can't see it, but there also are fluorescent microspheres. And if we turn off the ring light, that's visible light, and turn on the laser, then we can see the fluorescent microspheres are actually advected around by that turbulent flow with an effective diffusivity, if you want to call it that is vastly larger than the thermal diffusion constant that these 1-micron spheres would have, because they're really getting trapped in these eddies and moving around and essentially getting a much enhanced diffusivity. So this is one of the takeaway messages that tracer particles in this kind of turbulent flow get pushed around much more than you might expect because of the advection by the eddies and the jets. So when we first discovered this and quantified it, we did some elementary particle image velocimetry from the bacterial motion itself. And you can see very clearly that there are these regions of swirls that are on a scale of something like 30 to 60 microns, again, much larger than the individual bugs. And this was, at the time, basically, I think fair to say, the first experimental support for the prediction by Sriram and collaborators that long-range hydrodynamic interactions between swimming microorganisms of this type would destabilize a putative long-range ordered state that might have existed if you believed in the sort of dry dynamics like the Tumner 2 or Bitschek type models. Subsequent work by Santion and Shelley and others building on the original formulation that Tim and John Kessler found that in a kind of more, maybe I can say, systematic way from an underlying kinetic theory for these systems. Now, I say these particular organisms because what we understand now very clearly is that the important distinction between swimming microorganisms that must be made is between pullers and pushers. So bacteria, cell body, flagella behind, exerting a force on the fluid from the flagella and a force on the fluid from the cell body is a pusher dipole, whereas something like Chlamydomonas, which has its 2 flagella going like this, is the cell body behind the flagella forward. That's a puller dipole. And it's only the pusher dipoles which will, by theory, tend to produce these kind of long-range effects. And the essence of it you can see here in work that was done by my then PhD student, Knut Drescher, long ago. If you actually measure the flow field around individual freely swimming bacteria, it is a classic pusher dipole. So this is a heroic experiment involving studying tracer particles as bacteria swim around and referencing the PIV to the moving frame of the organism. And what you see is that there's flow out the front, out the back, and in from the sides. And that in from the sides will tend to make them cluster together in parallel and produce these coherent motions. So if you do a more sophisticated analysis of these kinds of flow fields in a more controlled way, so a very thin microfluidic geometry where the system is closed and over time the oxygen is being depleted, but the bacteria are still swimming until the very end, at any instant of time you'll see a flow field which is like this, but if you map out the vorticity, it looks a lot like a real turbulent flow. And in fact, if you quantify things by just, for instance, plotting the entropy versus the average energy, there'll be a squared length between them, and that's pretty much constant. As the bacteria use up the oxygen and the average speed is going down, these two things remain proportional to each other, giving you an estimate of the size of these eddies, something like 25 microns. Okay, so that's just to say the choice of Bacillus subtilis is in part because it's the system that we know produces collective behavior at high concentration, and we know a lot about its dependence on oxygen, et cetera. So for those of you who are new to Cambridge, you may think it's strange that I'm talking about experiments, even though we're next to the math department. But that's rule number one in Cambridge. What goes on in departments and the name of the department have nothing to do with each other. All right, so this is what my lab looks like, pieces of it. So we have, the usual wet lab facilities, diurnal growth chambers to grow algae on a day-night cycle, as you see here. We have various kinds of microscopes. Here's a light sheet microscope, not relevant for today's talk. Typical kind of microscope we use, high-speed camera, micromanipulation, optical methods of all types. So we wanted to develop a kind of controlled way of shining light on a system of algae, having them produce oxygen, and then watching the bacteria respond to that. And so, of course, for the paper that we wrote, we had to come up with a schematic. So I succumbed to the pressure from YASA to let artificial intelligence interpret our desires, and it came up with this. The idea, there's a little chamber and there's an eye that's looking down from above. It's very disturbing, but it's a little bit too abstract for our purposes. So this is what the real experimental system looks like. So the idea is we have a quasi-two-dimensional suspension of bacteria and algae at initially uniform concentration that we can control. And we bring in a photosynthetically active light as a shaft, just illuminate some small portion of the chamber. And we can look at this two ways, normal bright field imaging where we can see the algae, but we can also, because we're using fluorescently labeled bacteria, we can turn that off and just see the bacteria by their fluorescence. So we can view things with two different means of observation. And here's what you see when you look down on the chamber, a portion of the chamber. There's a region defined by the iris far outside the microscope stage area where the shaft of light will exist, and it typically shows up as a hexagonal pattern, and then the algae and bacteria are doing their thing there. So we discovered a very interesting and unusual phenomenon, which we're calling algal expulsion. And it looks like this. So at time t equals 0, you turn this shaft of light on, and then the algae that are producing oxygen photosynthetically, of course, are sitting there. These are non-motile algae. They're just little factories of oxygen. And of course, then the bacteria respond to this oxygen, and they chemotactically move into the eliminated region. And you can see that here. They pile up at high concentration. And interestingly, as they do this, the algae get thrown out of the region. And there's a vast depletion of the algal concentration. And then the bacteria, having done their damage, go back to where they were.
So the top is the bacteria and the bottom
is the algae. Yeah. So over time, the bacteria flood in and then dissipate out, and the algae get thrown out of the inside. So we call this algal expulsion. Yeah.
Two quick questions. Do you grow the species together before T0? Is it a co-culture from the beginning, or you just add them and then do the experiment?
No, it's a co-culture from the beginning. Yeah.
And is there a peak in the amount of oxygen that bacteria is interested in? So is it like high oxygen repel them also? Like
I'll get to that. There's a subtle point about how they respond. Yeah. Okay. Now we also discovered. Sorry, let me show you this video. I hope it plays. So you can see it's just animating those pictures, but there's this pile up, and then the bacteria dissipate out of the region, and the algae get thrown out. Now that's what we'll call type 1 expulsion. But we also discovered at much higher bacteria concentration what we call type 2 expulsion, where it turns out that not only are the algae thrown out, but also a significant number of the bacteria are thrown out as well. So they rush in, some of them stay for a while, but many of them are actually expelled to a region around here, just outside the illuminated region, where they pile up at very high concentration. Yes?
So why do you have octagon and not simply a sphere?
It's just the shape of the iris.
I mean, a sphere would be more natural, right? So,
You mean a circle? Yeah, well, it was the easiest thing to do. Yeah, doesn't matter. Yeah. Okay.
Sorry.
Yes.
Sorry. Is it an expulsion, or is it that the bacteria or the algae from the outside cannot get in because you have a flow that circumvents?
I'll show you. Yeah. Okay. So if we want to try to quantify this, we can, right? We know the fluorescence intensity of the bacteria. We can relate it to the original concentration field. So here we can show you, in the case of this type 1 dynamics, this is what the bacterial density looks like as a function of the distance from the center of this quasi-circle, with apologies for that. So the diameter of this illuminated region is 400 and some odd microns. So we're talking about scales very large compared to the individual organisms. And what you see is the development of a very strong concentration gradient of bacteria pointing toward the center. And over time, meaning tens of minutes, the algal density basically drops quite significantly. It doesn't completely get expelled, but drops by about a factor of 2, and they pile up on the outside. So they're thrown out. Now, in the case of this type 2 evolution, the bacteria indeed concentrate more in the center, but their very quickly overall concentration is decreased as they're thrown out to this region out here. And if you look carefully in this peak out here, these bacteria are almost immotile. So the ones in the center are moving fine, the ones at the edge are basically immotile. But still, the algal expulsion occurs. Now, when you look in this region, you can sample different parts of space and look at the mean square displacement of a given algal cell as a function of time, as a measure of its effective diffusivity. And what you discover is that as you increase time in this kind of measurement, there's a general increase in the slope of these curves. So there's some region in which it looks effectively diffusive. But as you wait longer and longer, it's becoming more and more super diffusive. And this is something that has been seen for several decades in these systems, that passive tracers actually exhibit super diffusion as a consequence of being advected around typically in these kinds of convective dynamics. But if you wanted to effectively define an effective diffusivity, say asking at what point the slope is locally 1, and plot that as a function of distance from the center, what you see is a very strong gradient in diffusivity from the center of the drop, the center of the illuminated region outward. Okay, so this is the basic set of observations, and we want to try to come up with some understanding of what we see. So we're going to do this in 3 stages. We're going to first ask about the oxygen dynamics, then we're going to ask about the bacterial dynamics, and then we're going to ask about the algae dynamics and try to put it all together and see what happens. So the oxygen is a fairly simple thing to think about, I think. So let's imagine this. We have oxygen whose concentration is C, bacteria B, algae A, so we're going to call this the ABC model. Eventually there'll be an ABCD model, but right now there's ABC. So how would the oxygen concentration evolve? Well, there's some oxygen diffusion constant, D sub C, with, depending upon some illumination pattern as a function of space, I of R, which in our case is a shaft, a top hat profile, there'll be some production of oxygen from the algae at some rate K+, and there'll be consumption of oxygen by the bacteria at a rate which, in the simplest case, would be proportional to the amount of oxygen in the system, but typically there's a kind of Michaelis-Menten structure to these dynamics, so the concentration, the consumption rate will saturate at high concentrations and only be linear at low concentrations. And this Michaelis constant Kc is a boundary between the two. So if you just want to get a handle on what's going on, you start with a uniform illumination everywhere and ask, how does the system balance? And you can find a balance between algal production and consumption such that there's a concentration C star that will prevail dependent upon the Michaelis constant and the ratio of these two rate constants, where A naught and B naught are the assumed concentrations of algae and bacteria. So provided this parameter K is less than 1, you can actually reach a steady state where the bacteria can consume all the oxygen that's being produced. But if your production rate is too high, then the bacteria can't keep up and you'll just flood the system with oxygen. But we're in the low production limit where the production and consumption are both limited in concentrations. Okay, so now you set up some light shaft with a radius r, and the natural thing is to scale time on the diffusive time for the oxygen to cross that region. Let's scale the space on that same distance, scale the concentrations in terms of their initial values over this C star. Then you discover there's a screening length in the problem, which is the square root of the oxygen diffusion times the characteristic consumption time of oxygen. And not surprisingly, if you have a top hat profile, you're producing oxygen in the center, it's going to leak out diffusively, and it's going to be consumed, and this will lead to an exponential decay in the outside on this length scale lambda. And so the oxygen dynamics in this rescaled units has this interesting form where this is a heaviside. And so if you just look in the linear problem, outside that domain, you have grad squared chi minus kappa squared chi, where kappa is the ratio of the shaft size to this penetration depth, which in our system is about 1 or 2. So basically that's just a Bessel function. And so if you plot this, you'll see that depending upon the ratio of r squared to lambda squared, you get top hat profiles where the concentration is pretty much at the C star or much less depending upon the size of the domain. Okay, so that's the picture we should have. And in fact, if you go back to the experiment, what you see pretty clearly is the appearance of a depletion zone just outside the illuminated region. And that kind of tells you the size scale of this penetration length. Okay, so that's basically what's going on with oxygen. Now the question is, what are the bacteria doing? And here we're just going to use the very simplest description that goes back to Keller and Siegel, who many, many years ago, 1970, first wrote down what we now accept as the basic model for chemotaxis of bacteria or anything else. And their idea is that the flux of microorganisms responding to a chemical gradient not only has a fixed law contribution that would look like the gradient of its concentration times some diffusion constant, but that they're basically climbing. How did that happen? We go into some Zoom? No, thank you. Okay. They're basically climbing the gradient of the chemoattractant that they might have. So with some constant G, which could depend on C, the flux is proportional to the amount of bacteria and the gradient of the concentration. So that's the simplest picture that you could have. And so your dynamics looks like diffusion minus divergence of G, B, grad C. In scaled units, it looks like this, where one cares about the ratio of the bacterial diffusion constant to the oxygen diffusion constant. Now, typically for this kind of random so-called run and tumble motion of bacteria, that ratio is not as small as you might imagine. So we're not talking about the thermal diffusivity of the bacteria. We're talking about the translational diffusivity associated with their stochastic motion. So this ratio could be something like 0.1 or so. And now if you take the bacteria and allow them to respond to that oxygen
Sorry, just one small bit. To use that effective diffusivity, you have to work on time scales long compared to the runtime or something. So I guess you're implicitly doing that, right?
Yes, although I'm going to go beyond that effective diffusivity shortly.
Ah, okay.
Yep. My point in saying that it keeps going up is to say something else happens on much longer time scales. Yeah. So what you see naturally is that for the bacteria to go in, they're going to create a depletion zone right outside. And then only on long enough timescales will they flow in from very far away to fill that in. But of course, the question then is at those very far distances, is there enough oxygen for them to do anything? Okay, so these two contributions are of the bacteria and the oxygen are fairly straightforward and maybe not controversial, but now let's ask what would happen to the algal dynamics. Okay, so I want you to remember what I showed you here, that when we have this collective behavior, which sets in at very high concentrations of bacteria, that passive tracers get advected around enormously compared to their normal diffusivity. But we have a very strong gradient in the concentration of bacteria. In the center of this illuminated region, it can be 10 times the concentration outside. Okay, so that says there's a gradient of activity. There's an inhomogeneous turbulence in the problem. And so after a lot of thought, we propose, we suggest, we hypothesize that there is an extra contribution to the algal flux besides what might come from an ordinary diffusion, a la Sriram's comment, that looks like the concentration of algae times the gradient of the bacterial concentration, because that's the gradient of the turbulence, essentially. So we're making the simplest assumption that that linear relation is all we need. Of course, you might say it's something higher, but this is the simplest one, A grad B. So that's a new term, or at least it's new in this context. And so then, if you plug that into the algal dynamics, allow for there to be a, let's say, ordinary diffusion constant that's sitting here, which is the one I showed you before by measurement of the mean square displacement. There's an extra piece now that looks like the divergence of alpha grad beta. Alpha's the scaled algae, grad beta's the scaled bacteria, which you could write this way, or if you expand it out, you get an advective contribution that's pushing the algae around, and you get a reactive term here, which is essentially responsible for the expulsion. Grad squared B, with a profile of bacteria like this, is negative. Right? So it's dropping the concentration of algae on the inside, and it produces this expulsion. Okay. So you can then try to actually fit the data to this using those constants. We have rough ideas of what they might be. And in this type 1 data, you see that you can pretty well accommodate the early behavior of the bacteria rushing in and the algae getting expelled. This is the eventual steady state profile of the algae. Okay, so that kind of explains the basic behavior of the type 1 expulsion. But now I want to bring you back many years to another experiment which reveals something very important about oxygen and motility and a phenomenon which you might call hypoxia.
Excuse me, do the usual scalings of the boundary layer apply, carried over to this problem?
Which boundary layer do you
I
mean?
mean, the expelled region, the thickness of...
Yes, it's pretty much consistent with that depletion length I was showing you. Okay, so when we first discovered this bacterial turbulence many years ago, there was a sort of subtle question about exactly what is going on with oxygen in the system as a function of the depth of the layer that we're interested in. So we came up with this is an old experiment, but important here. We came up with a simpler way of looking at the problem where you basically make a Healy-Shaw cell up on its end with a drop of fluid of this shape. You put a ring light outside the field of view out here so you can do dark field imaging to see very nicely the bacteria here. But you also have a laser that goes in so you can study the tracer particles as well. And so your camera can see either of those two things. And when you do this experiment with an initially well-mixed suspension of bacteria, what you discover within a couple of minutes, about 2 minutes, is that the initially uniform concentration of bacteria, which you see in the dark field here, evolves to have this very sharp boundary with apparently a depletion zone here and a higher concentration of bacteria at the top. And this is something that's been of interest in similar contexts for a long time, even going back to the work of Tim and John Kessler back in the 80s. Basically, what appears to be happening here is the bacteria are consuming oxygen. They are also oxygen tactic, but their motility is a very strong function of the oxygen concentration. And so basically, if you think about the concentration of oxygen from the top into the depth here, it's basically exponentially decreasing. There comes a point at which it's so low, they have no motility. And beyond that, they just have enough oxygen to climb the gradient and get into the region of more oxygen and make it to the top. And so there's this sharp boundary where they leave behind, unfortunately, all their brethren down here to suffocate while the few lucky ones make it to the top. However, it's not all so bad, because as soon as the bioconvection occurs, everybody gets stirred up, and so these get saved. But the important point is that there is a very clear motility transition under hypoxia, the lack of oxygen. So we asked ourselves, could this possibly allow us to understand this type 2 expulsion? And the basic idea is then to say, well, there are 3 species in the problem. There's algae, bacteria, oxygen represented by C, and there's also the dormant state, the hypoxic, non-motile state of bacteria. And we'll basically just construct a kind of population exchange model in which the dynamics of bacteria has a depletion that depends on the oxygen concentration and some characteristic cutoff value in this kind of monode formulation. And those same parts of the population reappear in a dormant state, and the dormant state is then another tracer particle, just like the algae. So it can be advected away in the same way. So it has those two contributions, the advective piece and the sort of expulsion piece. And if you do that, then you actually understand not only that the immotile ones get expelled, but also that they get expelled and the bacteria form a kind of barrier to the further diffusion of the algae outwards. So the two get trapped together in that outer layer. Furthermore, you can actually make a kind of spatially average model where you say, well, forget all the details of what's going on inside here. I've just got this illuminated region. I've got all this stuff going on inside, but most of the gradients are living at the edge of the domain. So let me just formulate this as a transport problem from a homogeneous concentration inside to something outside. And you can build this kind of compartment model. And what you discover, which you can also see earlier on, is that the one place where we can't seem to understand things very quantitatively is that while the algae get expelled over time, our prediction for the bacteria expulsion afterwards, when the algae are no longer there and the oxygen is dropped, is much too slow. So the real decay of bacteria back to the steady state is much faster than the model predicts. And we think that this could be a consequence of adaptation of the bacteria to the oxygen concentration. So at some point they just are not so sensitive to it anymore. And then they just say, well, fine, then I'll just diffusively leave. So that is what we think is going on. Okay, now, I keep calling this expulsion. We originally kept calling it flux expulsion because, you know, one of the things that happens when you sit in damp is you get expelled to different, you get exposed, you get expelled, yeah. You get exposed to different fields. And one of my great privileges in coming here was meeting and getting to know Nigel Weiss, who sadly passed away some years ago. But one of the things I learned, as everybody in damp learns, is the famous paper of his about flux expulsion from many, many years ago. And so basically our understanding of this is the following. You write down essentially the equation of motion of the magnetic field in a MHD setting. It's pushed around by the fluid velocity, and there's some dissipative term that's 1 over the conductivity of the medium. And if you just sit in a simple geometry where the B field and the velocity field are in the XY plane, and all you have is the vector potential A with a single component along the Z direction, then the dynamics of the A field is just an advection diffusion problem. And what Weiss did, and which Yasa redid using a slightly more efficient computer code many years later, is to consider a variety of different specified velocity fields and just watch what happens. And so the simplest thing is you have a single vortex velocity field like this, and you just let the system go. So you start with an initial condition where the vector potential is linear in one of the coordinates. That gives a constant magnetic field. Here are the field lines pointing down. And you just let this evolve under that equation of motion. And what you find, of course, is the field lines get all twisted up by the vortex, and the vector potential homogenizes inside the vortex, which means the magnetic field has been expelled. So this is flux expulsion by a single vortex, and there's an enormous literature on that. And I kept thinking, you know, that somehow there must be a connection between these two problems. I mean, it's the same kind of equation that we're dealing with, except that we don't have a single vortex. And then in further understanding the field, I came across this very important follow-up paper, Flux Expulsion by Inhomogeneous Turbulence. So my reading of the field is that after people thought about a single well-defined vortex, they said, yes, but you know, if there's a turbulent system and the turbulence has a gradient in its intensity, then things will be advected from the regions of high turbulent activity to the regions of low activity. In the context of the passive tracers I showed you, that would be like the particles are getting advected around in this turbulent system, and they move to the quieter regions, because they're always getting expelled to the separatrices between these vortical regions, and that will push them to the quieter region. So what Tao, Proctor, and Weiss did was a bunch of numerics and a bit of analytics where they said, you know, effectively there's some average vector potential component on a long scale, which obeys a diffusive equation like this, if you then ask about the magnetic field, the average magnetic field that follows from this A, it obeys an equation which is essentially identical to what we wrote down. It has an ordinary diffusion piece, it has this reactive term that looks like this, and it has an advective piece. So it seems like we've just kind of discovered, in the case of an experiment on this scale, pretty much the same mathematical structure as what is proposed in the case of MHD on vastly larger scales. I think that's kind of interesting. And it raises a whole bunch of really interesting questions about inhomogeneous active matter. So since I'm the only thing standing between you and lunch, I thought I would make this not such a long talk, and I'll just end there and dedicate this to the memory of John Kessler, who did so much for this field to get us all launched in studying collective bacterial dynamics. Thank you.
Thank you, Ray. Well, plenty of time for questions. Yes, Declan.
Well, thanks for your nice talk. You mentioned the convection driven by buoyancy, but there's also, in fact, convection driven by concentration differences, the Maragoni effects. And you have gradients in concentration at the surface. And did you estimate whether Maragoni effects would matter here?
So just be clear, in the original experiment long ago, we had a free surface. Here it's a closed microfluidic chamber. There's no air-water interface, if that's what you want to see. So there isn't.
But in the original one, I mean, I could imagine that Maragoni was matter.
It certainly can be, in particular because Bacillus subtilis, in particular, is known to exude surfactants.
Yes.
And so, indeed, there could well be Maragoni effects there. I don't think they've been considered too much. I'm looking at Tim, because it seems like things are dominated by the oxygen dynamics, but it could well be.
But I mean, you can calculate the Archimedes number to see whether it matters, and the Maragoni number to compare.
For sure. I mean, if you wait long enough, the surface becomes almost rigid from surfactants that are given off. So eventually it has to matter, for sure. Yeah.
So I just wondered, thank you for very nice talk. So I just wondered why you can treat turbulence at 2D or quiescent 2D. May I understand it is due to the uniform illumination from the
So,
side?
several things. First of all, the depth of the chamber is rather small.
Small.
Yes, so it's some multiple of the typical correlation length of the turbulent behavior that you might see, but it's not a real tall chamber. So we're looking at depth average quantities, if you want. And everything is very wide compared to those structures.
Yeah, but you mean the length scale in the vertical direction is very short.
Yeah.
But it may be very, gradient might be very large.
It's possible.
Then I just wondered, in magnet MHD case, we need a rotation or a magnetic field, something like that. Then I just want consider this uniform
I mean, one thing I would say is that actually one of the slightly tricky things in this whole discussion of bacterial turbulence is if you actually try to understand the length scales that come out here. I mean, experimentally, what you find is that the correlation length in the plane is not so different from the depth of the chamber in some cases. And so you can do all the fancy analysis you want mathematically, but you may be actually seeing a 3D confinement effect in terms of what's actually selected. So that may be the case for sure, but in some ways that doesn't matter so much for our way of thinking about the problem because we're saying there is a turbulent collective state, whatever its length scales, it may be set by the chamber, but whatever it is, it's producing this inhomogeneous transport. But it's an important point, absolutely. Yeah.
So this is a really wacky question, but I can't resist. You know, of course, the famous industry of Weiss et al. is fundamentally a kinematic problem, right? There's no back reaction.
There's no back reaction
but
in there.
Really in MHD, of course, there is, right? And And our distinguished chairman here and others have looked at that, and there are regimes you get into where you don't expel, where the back reaction is strong enough to destroy the vortex. So it begs the question in these, in these active matter problems, is an outcome like that lurking yet undone?
Okay, so not wacky at all. Brilliant, I would say. Sorry. So I pointed out that there are many useful mutants of this fellow. And as I was listening to, I was thinking, what's the equivalent of back reaction? Well, what you want is the thing that's being expelled is the algae. You want it to affect the flow. And so we deliberately engineered the system to be immotile algae, just little factories of oxygen. But on our to-do list is give them back their flagella. Right? So now the little factories of oxygen can move. And they will affect the flow. Now they're polar dipoles, so they won't necessarily have an equivalent effect of canceling out what the bacteria do. But that would be kind of along the way toward allowing some sort of back reaction. Yeah. And I mean, this is one of those things I often deal with, the question of what's your hypothesis. You know, if you're dealing with biologists, they'll always say, you know, hypothesis-driven research. And this was just, let's just see what happens kind of research. Right? And so the next step is also, I can imagine all sorts of scenarios, but let's just see what happens. Next week.
Tim, please.
I'm just remembering one experiment that John Kessler did, which was to trap an air bubble between two microscope slides in a fluid, and at T=0, and the fluid contained bacteria, Bacillus subtilis. And you set it at T=0, and then as time goes on, you observe the concentration of bacteria piling up at the exterior of the bubble. And the bit that I would just comment, yeah, that's it, yeah. And as it would be expected by what you told us about, I just remember one or two of the photos which I never understood, and nor did John, and we never made anything of, in which there appeared to be, whereas you'd expect the bacterial concentration to be a maximum next to the oxygen source and decay, but there appears to be a secondary maximum in the bacterial concentration field, and I didn't understand it, but maybe I should have talked to you about it a bit more. I had forgotten about it.
I know this observation well. John and I played together with it, and I always thought it was quite fascinating because there was clearly a kind of set of circulating turbulence, back and forth. And it could well be that if there is any, say, a toxic component to the bacteria, that they could be shunted out here. Maybe that's what's going on. I don't know, but it's worth a try.
Guillaume next.
Sorry, you might have said it, and I misunderstood, but on this matter of expulsion versus transport barrier, you have one or several predators, you have one prey. One might expect that you get into a case where you have a flow that prevents things from getting in or out. So is it really a matter within your illumination zone of expelling stuff, or things that cannot get in because they encounter a barrier and then the gradient doesn't work anymore?
Well, the algae are originally inside the illuminated region. They're everywhere. It's there's some statistical fluctuations, but it's a uniform concentration of both bacteria and algae. So they're starting out inside and ending up outside rather than not being prevented from going inside. The bacteria can do their thing and move in and move out. But the algae themselves have no motility They are only advected by the random turbulent flows and thermal diffusivity, which is very small. So I agree, there could be a predator-prey dynamics, and that's why I think, or something akin to that, and that's why I think adding back the flagella to the algae and giving them motility again could make a very interesting dynamics. But I don't know of any specific predator-prey-like interaction between these two particular species, but you could certainly choose a different set that would have something like that, if I'm following your question.
Yeah, no, I get it, but the algae, they can't move. That's fine. So
They're only randomly transported by the flows.
But bacteria, I guess I'm asking as well, what is the typical time scale? Because the bacteria, they can die if they don't have oxygen anymore. You know, they just consume everything. No algae, they die. So in the end, you have a bigger concentration outside just because maybe the ones inside have died. Is it?
Yes, but in for instance, in the type 1 expulsion problem, the algae that retreat back are still motile. Okay, so the bacteria that retreat back are still motile. So there's still enough oxygen being produced by the algae in the center for a region to be animated, but not at the higher bacterial concentration. So I always think of this a little bit like sumo wrestling. I mean, if I understand the rules of sumo wrestling, you wrestle and wrestle, but if you step out of the ring, it's over. And this depletion zone means of oxygen consumption means the oxygen concentration is dropping precipitously outside this region. And so basically, if the algae step out of there by being randomly transported, they're no longer producing oxygen. And the bacteria then are very quickly in this hypoxic region. And so their motion stops as well. So everybody just stops when they go outside.
Yeah, I was thinking to the first question you asked about the B12 thing there. So do you have any ideas of the consequences of this expulsion stuff and so on?
I don't think this has a direct bearing on that because, like most of these kinds of questions, you get lost in some tangential directory because there actually are specific physical interactions between the algae and the bacteria that are...
Do they have time to do it? Or?
Yeah, and in some ways also, we're talking here about symbiotic dynamics on the scale of tens of minutes, whereas that kind of question is on a much longer time scale, days, weeks, whatever. So in some ways, this is a sort of new aspect of the symbiotic dynamics to think about it on the scale of set by motility and diffusion rather than the more ecological timescales.
Okay, I just had one comment. So in the Weiss paper, even though it's just dA by dt, u dot grad A, his arguments rely crucially on the fact that that B is grad A, if you like. Whereas if you start from like the temperature equation, which looks just the same, you don't get the same answers. So it's sort of interesting that you've gone.
Yes. I mean, it is subtle.
It's become a magnetic analogy.
Yeah.
Because the A in that problem, it really matters that the B is.
Exactly. Yes. And so here it's the scalar itself that's being expelled, rather than the curl of the scalar. Right?
Yeah. But if you took the temperature equation and then do the standard mean field approach on that, you get a different answer in that case, interestingly.
Yes.
Yeah. Okay. Okay. Well, first of all, let's thank Ray again.